Hi, today I will be talking about a bacteria which is named as Streptococcus. Now we have more than 100 species of Streptococcus. This group of bacteria, it is gram-positive in nature. When we do the gram staining, which is the differential staining, we can divide the bacteria into two groups. So in that, this Streptococcus group comes under gram-positive. It's a cocci because the shape is spherical. So when you see the bacteria under gram staining, what you see is gram positive, which is purple. When you do the gram staining, which is gram positive, will appear as purple. So this bacteria appears as spherical, purple, and joined as a chain. So it's a long chain. Sometimes you can see when you take the throat swab, when you take a sample from your throat, you can see this bacteria in small chains, long chains. So we have varieties of organism, but as a normal flora, you can see them in our body. So this bacteria is a common commensal in our body, but some of the species can lead to infections. So here we will be dealing with those bacteria which can cause human infection, even though they are present as normal commensal. So before going to the organism, the Streptococcus bacteria, as we have many, we are classifying into different categories. So this broad classification first is based on the oxygen requirement. When the bacteria grows in a culture medium or when it grows outside, whether it requires oxygen or not, based on that we have two groups broadly classified into aerobes and anaerobes. But here the classification is, we are classifying majority under aerobe and facultative anaerobe. Aerobe is an organism which requires oxygen for their growth. Facultative anaerobe means they are actually anaerobe, they cannot survive in the presence of oxygen, but they may survive because it is facultative. So facultative anaerobe and aerobe is one category. The second category is obligate anaerobe. Now anaerobes are bacteria which cannot survive in the presence of oxygen. Obligate means they are very strict regarding this. That means they can die in the presence of oxygen. So we have only one example under obligate anaerobe, that is Streptococci. The other group, aerobe and facultative anaerobe, as we have many, we are again classifying based on the hemolysis. It's a property which is seen on blood agar. So when we grow the bacteria on blood agar, you divide the bacteria into three groups. One is alpha hemolytic, beta hemolytic, and gamma hemolytic. This is based on the hemolysis. It's a property which you can observe only on blood agar. So when the bacteria grows on blood agar, if the hemolysis is partial, it's not complete, then you call it as alpha hemolytic streptococci. So we have a group of organisms coming under alpha hemolytic streptococci. You call them as viridin streptococci. Why viridins? Because the word viridis means greenish. Then that means when these bacteria grow on this medium, culture medium, blood agar, you see a greenish discoloration around the colony. That is because of the partial hemolysis. So we have any examples under this, Streptococcus mitis, mutans, and salivarius. There are many. These organisms are seen in the oral cavity as a commensal. Now what happens is, normally healthy person, they don't get any kind of infection with this group. But in some conditions with predisposing factors, suppose you have a congenital heart issue or anything, these organisms again can cause subacute bacterial endocarditis. So it's a clinical condition seen in some people because of the normal organism present in your oral cavity. Again, these organisms can become pathogenic. You can consider them as pathogen when you have low oral hygiene. For example, when you are having less oral hygiene, it can lead to dental caries. So dental caries and subacute bacterial endocarditis become the only pathogenic condition caused by viridin streptococci. So they are alpha hemolytic. They grow on blood agar with greenish discoloration. The second group is beta hemolytic, and majority, as you see, it comes under beta hemolytic. Streptococci are coming under beta hemolytic streptococci. Now what is beta hemolysis? It's complete hemolysis. You see a clear zone around the bacterial colony when they grow on blood agar. So again you can see that it is classified into various groups because majority of our organisms come under this. So based on the serological typing, that is when you collect the serum sample from the patient, you can divide these bacteria into different types, that is called as serological groups. So, serological groups are based on a kind of polysaccharide which is present in the cell wall of this bacteria. All the Streptococcus which belong to beta hemolysis will have this carbohydrate polysaccharide which is present in the cell wall. Again, based on that, we are classifying into different groups. This is named after Lancefield, Rebecca Lancefield, and currently we have 20 Lancefield groups. Now, Lancefield groups are named as A, B up to V. In between I and J, they removed it. So, now we have only 20, though it starts from A and ends with V. So, these are called as Lancefield groups. Okay. Now, coming to the third one, that is gamma hemolysis or gamma hemolytic streptococci. Truly speaking, it is not hemolytic, it is non-hemolytic. When you see no change when this bacteria grow on blood agar, you call it as gamma hemolytic or non-hemolytic streptococci. We have a group that is enterococci which comes under this. These enterococci again classified based on the physiological and biochemical properties which we will be dealing it later. Now, coming back to the beta hemolytic we have Lancefield grouping. In that group A we are going to explain in detail. So, that because this is one of the most common pathogen under Streptococcus species. So, this group A which is coming under Lancefield group A is known as Streptococcus pyogenes. Now, Streptococcus pyogenes is our pathogen which we are going to take in detail. In that again we have more than 100 types. So, how do we classify? We classify based on the protein which is present in the cell wall, that is known as M protein. Is a protein. Based on that, what type of M protein is present? We have different types, that is known as Griffith type. It is named after Griffith. So, we starts with number 1. It is currently we have more than 100 types of Griffith Streptococcus pyogenes. So, these are called as serological typing or Griffith type. So, we need not worry about that part because we have many groups which we normally for a patient we don't identify which is the Griffith type. Usually for a patient it is very important to identify whether this bacteria is Streptococcus pyogenes or not, because though it is a normal commensal in the oral cavity or the throat, it can lead to mild infection or sometimes life threatening infections. So, coming to the Streptococcus pyogenes which is group A Streptococcus, otherwise we call it as GAS, that is group A Streptococcus species. Now, usually we study the organism based on its morphology, how it grows on culture medium, what are the biochemical tests we can do, how is the virulence factors, then we study about the pathogenesis part and the lab diagnosis, then finally how do we prevent the infection and how do we treat the infection. So, going to the first part, that is how the bacteria look like. We know it is a Streptococcus bacteria, that means it must be a gram-positive cocci with arranged as chains, and other features are not there. It is non-motile, non-sporing, no other important morphological features except that when you do the gram staining it appears as violet, you call it as gram-positive. It appears as spherical shape, you call it as cocci, and these cocci will be arranged in chains. So, that means gram-positive cocci in chains. Coming to the cultural property. Cultural property means how you are going to grow the organism in your culture media. Artificially, you can grow the organism. When you collect the sample from the patient, what we can do is we can grow the organism in the lab with provided we give all the required for the growth of the organism. For example, this bacteria is aerobe and facultative anaerobe. So, you need to provide oxygen for their growth, air for their growth, because it requires oxygen for their growth. Temperature required is 37 degree and pH is 7.4, optimum pH and temperature. There is a temperature and pH which is required for the best growth of the organism. Then this bacteria grows only on medium which contains blood and serum. That is why we grow the organism on blood agar. As we discuss the in detail how do we classify, we saw the Streptococcus pyogenes comes under beta hemolytic streptococci. That means it shows complete clearing. As you see, there will be a clear zone around the bacterial colony that shows beta hemolysis. And if you want to see this beta hemolysis enhanced, what you can do is you can grow the bacteria in the presence of 10% carbon dioxide. That will lead to the better hemolysis. Coming to the test, which are the tests which will differentiate this bacteria from other bacteria? We have varieties of biochemical reactions based on our requirement, whether you are differentiating from Staphylococcus or whether you need to do the species differentiation. We have sensitive test, we have many biochemical tests. So, the first one is catalase negative. That means when you do the catalase test, it is a biochemical test to differentiate a similar organism, a bacteria which morphologically look like Streptococcus, that is Staphylococcus. This bacteria is catalase negative. Other tests which are important, it is insoluble in 10% bile, it hydrolyzes PYR, pyronidonyl, that is a test to differentiate it, and ribose sugar is not fermented by the Streptococcus and its sensitivity to bacitracin. So, this is how we differentiate the bacteria based on the biochemical test. So, we have discussed about the morphology, culture and which are the biochemical tests in this class. Thank you.